Welcome everybody to the April meeting of our NIAID Adjuvant Webinar Series. So I'll make a quick note that we very recently broadened the invitation list for this series. So we're going to include some more adjuvant researchers, both from academia and industry. So if you're new to the webinar series and this is your first time, welcome. So today we're going to be hearing from Matt Booty, who is the lead scientist at Spring Discovery. And Matt's going to be telling us about some new technologies for adjuvant discovery. If you have any questions during Matt's talk, please put them in the chat box. And we'll have some time at the end of the talk to answer these questions, as well as any additional questions that the audience may have. And then one more note, if you want to request a copy of Matt's presentation, you can send me an email and I'll drop my email in the chat so you all have it. So I think with that, we can get started, and I'll pass it over to you, Matt.
Sounds good. Share my slides. All right. Well, first of all, thank you for the invitation to share our work with you today. And I'll be talking about the discovery of novel adjuvants, and in particular, small molecule adjuvants. And at Spring, the approach we're taking to achieve this is to apply recent advances in computation, particularly deep learning, and applying this to high-dimensional phenotypic drug screens. And I'm going to walk you through one of those phenotypic drug screens today. Just a little bit about Spring as a company. So broadly, we are focused on aging and age-related diseases. And we were founded on the belief that this is a pretty complex space biologically, and we really need techniques like machine learning and specifically deep learning to accelerate drug discovery in this area. What's unique about Spring Discovery is that we're a pretty even split of wet lab biologists and computational experts. So really every stage of the experimental process, someone like myself, who's a biologist, is working side by side with my computational counterparts to build these experiments and to really build models to understand and interpret the data. As I said, Spring is focused on age-related diseases, but as you'll see, the platform itself is quite flexible. It can be used to discover targets and compounds to treat a variety of disease areas. And in particular, the focus today is discovery of these adjuvants, or more broadly, innate immune agonists. And as you can imagine, these will work in any individual, regardless of age. And in particular, I'm going to show you data from donor PBMCs today. The bulk of the data I'm showing you is actually from middle-aged donors. So within the umbrella of aging, there's obviously a lot you can study, but we are particularly interested in the aging immune system. And all of the changes that occur to the immune system as we age are broadly fall under immunosenescence. And immunosenescence encompasses both the changes in the innate and the adaptive side of the immune system. Certainly on the adaptive side, there are many changes as we age: decreased thymic output, a dearth of naive T cells, decreased responses to things like vaccines, or an ability to mount responses to infectious disease. There's a lot to research there, but we've chosen to focus on innate immunity first, and that's because we believe innate immunity offers a lot of opportunities to treat age-related diseases. I don't have a lot of time to go into it in depth today, but we do have active programs looking at decreasing inflammation, in particular inhibiting things like the inflammasome. And as we age, there's just a basal increase in inflammation. So it's interesting from that aspect. This is often termed inflammaging. But if you take decreasing inflammation in a more targeted fashion, there's a variety of disease applications. And we've actually undertaken a clinical trial in COVID-19 looking to limit some of that pathological inflammation associated with severe COVID disease. And we're expanding this now into new disease indications. But the subject of the talk today is on the other side of things, increasing targeted forms of inflammation, activating the innate immune system for the purpose of building vaccine adjuvants or other therapies. And broadly speaking, we believe activating innate immunity in the right ways has a lot of potential for therapeutic intervention in a variety of diseases. For vaccines, it's well documented that as we age, our response to vaccination decreases for a variety of reasons. This is seen especially in the seasonal influenza vaccine. And we also know what the solution to this problem is in a sense. That's better vaccine adjuvants. A more targeted engagement of the innate immune system can elicit productive inflammation and give you better outcomes for vaccination. But because we're looking for small molecule adjuvants in this case, or innate immune activators, the applications expand beyond that. And in particular, there's a lot of focus right now, obviously, in oncology, looking for compounds that can change or alter the immunosuppressive microenvironment of the tumor. We have quite a bit of data on actinic keratosis, so I'll just talk about this briefly. Keratoses are neoplastic lesions that form on the skin as we age. This is the result of UV exposure, and in many ways, this is a first step towards skin cancer, particularly cutaneous squamous cell carcinoma. What is interesting about actinic keratosis is that there's a variety of ways to treat this topically, but it's been shown in the clinic that innate immune activators can actually lead to lesion clearance, particularly imiquimod, TLR7 agonist, can produce inflammation that clears these lesions. And we actually have mouse model data here showing that some of our compounds are able to do this as well. And then finally, a more lofty goal, something to think about for the future, is the idea of infection prevention. I'm speaking to things like the use of BCG vaccination, for instance, that can induce states of trained immunity that can boost one's ability to fight infections, particularly in elderly populations. So for our discovery efforts, we focus on two key data types. That's high content imaging. So a lot of what I'll focus on today are models constructed on images taken through microscopy, but we also utilize proteomics at the well level as well. All of this information gets fed into our computational platform to ultimately construct what we term a phenoprint. And a phenoprint is essentially a computational model of a disease state or a cellular perturbation. So you can build a phenoprint to discriminate things like inflammasome activation or specific forms of inflammasome inhibition. You can also build models that predict young versus old. So looking at cells, you can build models that can look at images and make predictions about the age of the donor from which those cells came from. The focus today is on innate immune activation, and here we take a variety of reference innate immune activators, stimulate cells with these activators, and generate these phenoprints so that we can now discriminate those forms of activation and begin to understand how our experimental compounds relate to that form of inflammation. Getting more specific with the types of data, for the imaging side, the data I will show you today was obtained using high content imaging or high throughput confocal microscopy. This is performed in a 384 well plate. We capture multiple fields of view per each well. So it's really large amounts of imaging data, terabytes of imaging data that get fed into the models. And then for the proteomics side, we're doing well level proteomics. So essentially taking the supernatant and in this case, looking with a focus on cytokines, chemokines, and in some case, shed cell receptors. For the imaging, we can do anything with the platform that you would do with confocal microscopy. So we can certainly stain with specific antibodies for specific proteins, but the bulk of our data comes from using organelle-specific dyes. And what we're trying to accomplish here is to quantify really broadly cellular morphology. And so these dyes stain a number of organelles, including the nucleus, cytoskeleton, mitochondria, cell membrane, and Golgi. And what we're looking for is with our perturbations, with our innate immune activators, our reference controls, how is the morphology of these cells changing? How is the composition of these cells changing? And we use this information to build our models. Broadly, this approach is known as cell painting. It's been used in cancer drug discovery. It's used quite frequently with cancer cell lines. But here specifically, we're actually using unfractionated PBMCs. So we take PBMCs from multiple donors, usually using many donors for our screening campaigns, and we plate these in the 384 well plates. And the PBMCs are being stained with these morphological dyes to gather information about the cells and how they're changing. And I'll get into it a little bit more later, but even just using morphology alone, we're able to build models and have the ability to discriminate different cell types, lymphocytes from monocytes, et cetera, using morphology as the basis of discrimination. There's a variety of reasons to take this approach. One of the big advantages of using high content imaging with this proteomics approach is really just the scalability of the platform. We're able to screen tens of thousands of compounds relatively quickly and at relatively low cost. So the data I'm about to show you on some of our computational models comes from a screen that was run with PBMCs. These are plated in 384 well plates and stimulated overnight with a number of conditions. I'll use a broad category. Basically, these are our controls or reference innate activators. This is going to be marketed vaccine adjuvants and also a few more directed TLR and NLR agonists. With that, we're also screening unknown compounds, and we get these compounds for this screen from a drug repurposing library. So the compounds we're looking at have been largely approved for other indications, and with them comes some form of annotation. So when we get a hit in the screen, we know what the primary target for that compound is, and we can then can build hypotheses for how that compound might be inducing the inflammation we're detecting. You stimulate the PBMCs with these conditions overnight, and then data acquisition occurs. So this is the staining and the acquisition of the high content images, as well as harvesting the supernatant for the proteomics analyses. And it's all of this information that gets fed into the computational platform to begin to understand how our reference innate activators relate to each other, and more importantly, how our experimental compounds relate to those reference controls and looking for compounds that induce inflammation. A little note about the various reference innate activators we used. Here we have marketed vaccine adjuvants. So this includes AS01, which is a liposomal formulation containing MPL and this interesting saponin, QS21. We also included AS03 and MF59. What I like to point out about these two in particular is that AS03 and MF59 are fairly similar in terms of their composition. They're not identical, but similar in the sense they're both squalene oil and water emulsions. And one of the key differentiators is really that AS03 contains this extra vitamin E derivative, which further modulates cytokine and chemokine production. And I should note here that we just recently entered into a research collaboration with the Bill and Melinda Gates Foundation, and we're expanding the number of adjuvants that we're examining in our platform. In addition to the marketed vaccine adjuvants, we can stimulate with any number of known innate immune activators. For this screen, I'm going to be showing R848, which is TLR7/8 agonist, CpG, poly-IC, LPS in combination with nigericin, which is going to give you both TLR4 activation and also inflammasome activation. There's actually quite a bit of pyroptosis here as well, which is important later on. You'll see we quantify cell death within these images as well. And then finally, cGAMP, which serves as our stained agonist. So you take the PBMCs, again, multiple donors, stimulate them overnight with these various conditions, and then perform imaging looking at the morphological and compositional changes that come with those stimulations. And I'm showing example images here for each of the various conditions. And if you really take your time and stare closely at these, you can start to make some associations between changes in the images and the various stimulation conditions. But by using large amounts of imaging data across multiple donors, we're able to build algorithms on these images that can discriminate the various stimulation conditions. And we start by understanding how the different conditions are different from each other, and then we will begin to understand how our experimental compounds relate to our reference innate activators. So this is a confusion matrix. It's a way of visually representing the performance of your algorithm for discriminating these various conditions. And what we've done here is essentially taken the experimental data. We took the bulk of that data and have used those images with the annotations to build models. So we build algorithms that are able to look at these images, make associations with features in those images, and discriminate the various stimulation conditions. And for now, I'm just focusing on imaging data alone. We can incorporate the proteomics into this, but for now, let's just focus on the images. So you do that with the majority of your data, but you set aside a small amount of those images to serve as a test panel. Basically, these are images that the algorithm has never seen before. And once you're convinced that your models are relatively robust, you then test the system by showing those images to the algorithm and seeing how good it is at classifying the images it has never seen before. And basically, each number in this matrix represents the predicted label that the model has called the condition as versus versus what the true label was based on how we designed the experiment. And if you look across the diagonal, what you're seeing highlighted in purple here are the correct calls. So the number of times the algorithm correctly predicted what those PBMCs were stimulated with based on the imaging data. So for instance, in this case, we had a lot of DMSO samples, 951 correct calls. The algorithm was able to predict our negative control in this case. Occasionally, there are some misclassifications here. You can see predicting poly-IC or R848 when in fact it was DMSO, but overall the accuracy was very high at 99%. As you go through each of these conditions, you'll see that the accuracy changes somewhat, but tends to remain relatively high, usually around 90% or above. And I'll call your attention again to ASO3 and MF59. Because these adjuvants are compositionally similar, we were interested to see basically would the machine get confused as to what the PBMCs were stimulated with. And you can see the classification accuracy for ASO3 is quite high at 99%. The performance for MF59 is a little bit lower at 88%. You can see some of those calls. The algorithm was thinking this was DMSO when in fact it was MF59. But what I find most interesting about this is that it's really pretty rare that ASO3 and MF59 are confused just using these images alone. And then the final bit I'll call your attention to, you can see here that our performance for CGAMP was lower relative to the other conditions. This is something we have actually corrected in subsequent runs. It's basically the concentration of CGAMP that is used. And so as you increase the concentration, as you would expect, you're going to get a bit more inflammation, more dramatic morphological changes. And so the models become robust. And we see that with a number of conditions. And you can really build these classification models on any number of things. We've built them, for instance, on cytokines as well. And there you can see a very nice dose titration that with increasing concentration, increased perturbation, the performance of the model increases. So this is a start to how we begin drug discovery. We can see, basically if we have compounds that get classified similarly to one of these reference controls, but it gets really interesting when you start to look at this in multidimensional space. And so this is a UMAP, which is a dimension reduction technique. It's somewhat similar to a t-SNE, basically just enables you to look at multidimensional data across two axes. And this is where we really can begin to infer relationships between these reference innate immune activators. So I'll call your attention again to ASO3 and MF59, similar compositionally, and that's actually reflected in the data here. You can see they are adjacent to each other, suggesting a relationship between these two vaccine adjuvants, but note that they are quite distinct, very little overlap. So the model is able to discriminate them, but also reflects the similarity of these vaccine adjuvants in the data. As we move across this cloud, you'll see next to that is positioned ASO1, another vaccine adjuvant, again, distinct but nearby. We have the sting stimulation here that's kind of mixed in with the ASO1, followed by poly-IC, which again is related but quite distinct over here. And the list basically goes on. You can begin to infer these relationships. RA48 and LPS will both target TLRs. They are close by, but again, very distinct in terms of the clouds that they generate. And for reasons we're still exploring, CpG in our system always seems to be quite distinct in terms of where it falls in these projections. These other clouds that I'm not talking as much about are actually AK therapies. So I mentioned actinic keratosis earlier. What's interesting to point out about these is they are not innate immune activators per se. This is why, most likely why they're not located adjacent to any of our reference innate immune activators. But you can see all of the samples for a given simulation hang quite closely together, but again, not associated with that innate immune activation phenotype. So what we do when we screen is that we can now layer on experimental compounds into the space, and you can begin to look at these data and understand how an experimental compound might be related to these various activators of the innate immune system, looking for compounds that will engage inflammation in the ways that you want. And that's really the fundamental basis of how we screen for these adjuvants or innate immune activators. The basis is to use these unbiased models. So these we build signatures and scoring metrics based on adjuvants, TLRs, et cetera. This enables you to sift through the compounds and look to see what the model is saying of the experimental compounds is most similar to your known activators of innate immunity. You then pair that with these targeted features such as the proteomics. In this case, the proteomics serves as almost almost an inbuilt orthogonal assay. Are you getting the productive forms of information that you would expect based on your unbiased models? And then finally, because we're taking imaging data, there's a lot of information you can gather from the microscopy images themselves. And we've built a number of systems to automatically quantify many features throughout these images and throughout the data. And I don't have time to go through every single one. There are actually hundreds, but I'll give you a few examples of some of the information you can pull out of just the images alone. So the first targeted feature to cover is cell composition. And we've been able to train models to discriminate various cell subtypes, in this case, just using these morphological dyes. You test this by using antibodies specific for the subtypes, but using morphology alone, we can quite easily discriminate lymphocytes and monocytes. As you can see, they're quite distinct morphologically. And even within lymphocytes, you see some specific phenotypes here. We're terming this cytotoxic lymphocytes. These are polarized cells, could be a CD8 or an NK cell. And then within monocytes in the myeloid population, there's a lot of morphological diversity as these cells differentiate, and you're able to capture that using this imaging technique. So monocytes in particular, when they're activated with TLR agonists, for instance, you can get an activated phenotype that you capture in these images, as well as macrophages and dendritic cells, which tend to be more rare in our PBMC populations, at least at this 24-hour time point or overnight time point. But you do see these morphologies within the PBMCs themselves as well. Fortunately for us, and rather intriguingly, death, at least cell death, has pretty distinct morphologies, and we're able to capture this as well, and it becomes a component of that composition that we generate for each of our conditions. So here I'm showing some dying macrophages. Apoptosis, of course, has very distinct morphology. You can see the blebbing associated there, and also earlier forms of cell death or cells on the way towards death, like pycnotic cells where the nucleus is becoming condensed. So just using this as an example, you can get compositional information on all of your compounds and all of your controls and begin to understand how various stimulation conditions are affecting composition in the system. And I'll call out STING here, which seems to lead to an increase in dying lymphocytes, something we see with some frequency. Basically, as you get a large amount of cytokine production, in some cases, you can get lymphocyte death there. Continuing to more specific features on individual cells. So we start with that cell type classification, and then we can build models within different cell types. So you can look specifically within lymphocytes or within monocytes. And here I'm highlighting mitochondrial dynamics. This feature we find interesting because it gives you some insight into the energetic state of the cell. And we really note two distinct morphologies in mitochondria that we quantify within our system. The first is what we're calling a reticular network. These are elongated mitochondria, perhaps arising from fusion events, or also just aggregated mitochondrial masses. You could associate this with more efficient energetics, and in some cases, even fatty acid oxidation. When we look across the reference innate immune activators, particularly the adjuvants in T cells, we note some distinct and stark changes here in terms of the formation of reticular mitochondrial networks in T cells, with ASO3 and MF59 seeming to have the biggest impact here. On the other side of the spectrum for this phenotype would be a fragmented network. This is more diffuse staining. You see individual mitochondria spread throughout the cell. This can be associated with forms of cell stress. Generally, fragmented mitochondria are associated with glycolysis, so faster energy production, but less efficient. And here I'm highlighting some monocyte data looking at fragmented mitochondria, again, with STING being one of the outliers, showing an increase in fragmented mitochondria within this population of cells. Physical cell-to-cell interaction is another way to gather information here. The way we've designed this particular experiment, we don't have T cells with the ability to recognize cognate antigen, but one could imagine building models to look for T cell interactions with APCs, but we see a variety of interactions. So lymphocyte with lymphocyte, lymphocyte with monocyte, and monocyte with monocyte. And even the monocyte aggregation, we've noted changes across a variety of conditions. And if you look through the literature, there's reason to believe this has biological consequence. Particularly aggregation can influence things like activation state and cytokine production. And again, calling out STING here, we have a monocyte-monocyte interaction score showing an increased aggregation of those monocytes following overnight stimulation. I won't keep going through the entire list of features, but just to highlight a few final ones. We have looked at antigen uptake. You can do this by including fluorescent proteins or dextrans into the system and basically looking where these things localize within the myeloid cells in your assay. Proteosomes are something we see quite frequently. These are puncta of actin, generally associated with cell motility, but can also be associated with antigen presentation. Dendritic cells are not super common in PBMCs, but we do note a population in all of our donors, and you can actually quantify and look at the dendrites that form within those cells. And then a little bit less relevant to this overnight assay that I'm showing here, but something worth pointing out is that macrophage polarization is associated with quite distinct morphological changes, and we can build very robust models of M1 and M2 macrophage polarization. This would more ideally be suited for experiments where you're taking that differentiation out over a number of days, but just another example of a feature you can build to classify and look for changes within your cells. So all of this information goes into the drug screen. Again, this was a library of bioactive compounds. I'm going to show you data from a screen of about 10,000 compounds. We're looking at the way those compounds affect phenoprints relative to our reference innate immune activators. And then we go through to rank these compounds looking for what we deem most interesting. And for us, the scoring and ranking is a dynamic process. So you're able to weight things based on what you think is important for your particular application. For instance, if you want a STING-like compound, you can prioritize STING similarity in your modeling, and you could even penalize for things. So if you wanted to penalize certain forms of cell death or penalize the production of IL-6, for instance, you could add that into your scoring. And then finally, I will show you some information from orthogonal assays that we've performed downstream of the screen. So it's a lot of information to sift through. And one of the things we've done to enable our scientists to really wade through all of this data is built a tool that we term the MegaMap. And the MegaMap is an online application that enables us to sort through data and prioritize what we find is interesting. So I'm just showing a still snapshot of the MegaMap here, showing our reference innate immune activators. Each row is either a compound or a reference control, and each column is a different feature. So here's a cytokine subset. You can see how this changes, or cell composition. It's red and green here, looks like a heat map, but it's not actually showing the degree of change. It's just showing significant, with green being a significant increase and red being a significant decrease. And it's this system that we use to sort through our data. And just to give you a sense for what it's like, it's fairly dynamic. You're able to sort compounds in any way that you would like. So here I'm highlighting ASO1 similarity, looking at experimental compounds that ranked highly in our models for similarity to this vaccine adjuvant. And then you can go through the individual features and see how a compound changes or doesn't change a particular feature. And as you see me highlighting over, there's tooltips here, so you can get more granular as you go through. So for instance, if I go deep into the cytokines and maybe want to know how CXCL10 is affected, here I'm just showing the controls, but you can see how it's increased with exposure to various innate immune activators or other therapies or experimental compounds. And then on that dynamic scoring side of things, you have the ability to basically go through and decide what features you want to penalize or bring to the top of your screen here. So for here's an example of a score where we were penalizing apoptosis, so trying to surface compounds with adjuvant similarity, but minimal amounts of cell death. And so because we used an annotated library, we happen to have known TLR7 and TLR7/8 agonists in the library that went in as experimental compounds. So as an initial sanity check, one of the things we did was go into the MegaMap and see how these known TLR activators performed in the system. You know, did the model call them out for their similarity to R848, or did we capture them otherwise? And we were delighted to see that 6 of 7 of these compounds were flagged for their similarity to R848. I'm showing that here in this static shot of the mega map. So here, green showing significant scoring for this TLR7/8 score. So we grabbed 6 of the 7. We did not capture one of these. I will note that this is a TLR7 agonist, and the reference here is a TLR7/8. So there is a biological difference there. But we were actually able to capture this compound through other means, and that's based on a custom scoring system, again, weighting features that we thought would be associated with these activators. And again, the cytokine data here, what I find interesting is that, this was done purely with the images. The cytokines serve as a nice orthogonal assay demonstrating that when, it's called to be a TLR7/8 activator, we're getting the cytokine profile that one would expect with these particular compounds. Diving into an experimental target, I'm really just going to focus on one today. Unfortunately, I can't disclose to you what the target is, but what you're looking at are a number of compounds that target the same protein. And I'm highlighting these in particular because they had some similarity to ASO3, no ASO1 similarity. Often that correlated with a similarity in MF59. So we were initially interested in these compounds for that reason. They didn't have the cytokine profiles exactly like a vaccine adjuvant, but we did note a pretty distinct IL-1 beta signature. And in fact, some of these compounds were the most potent inducers of IL-1 beta that we found at the screen. So we decided to focus on some of these for orthogonal follow-up assays. One of the things I will point out about these drugs is these were actually developed as cancer chemotherapies. So this drug class was developed and designed to slow or inhibit cancer cell growth, but by screening on PBMCs, what we're seeing is a signature of inflammation and an elicitation of cytokine production. So this is an orthogonal assay looking at the ability of these compounds to mature dendritic cells. So we've taken monocyte-derived human dendritic cells, exposed them to some positive controls, here LPS and poly-IC, or experimental compounds, drug 1 and drug 2 that I highlighted in the previous slide. These are dendritic cells. They've been differentiated from monocytes. This is the flow data gating on them, looking at CD86 as a marker of maturation, so upregulation of a co-stimulatory molecule. As you can see with media alone, these are DCs. They express CD86. But as you add a positive control like LPS or poly-IC, you get a nice shift in upregulation of CD86. And likewise with these drugs, drug 1 and drug 2, again, targeting the same thing, we saw a nice increase in CD86 expression on these DCs. And this suggests that these drugs, these small molecules, are able to mature dendritic cells much like a PAMP would and perhaps like a vaccine adjuvant would. And so we decided to focus on one in particular, drug number 2, and move it into an in vivo model to see if it could serve as a vaccine adjuvant. This is a mouse model of intramuscular influenza vaccination. Specifically, we're using influenza nucleoprotein as a model antigen. And basically, you take the vaccine, which would be nucleoprotein, either formulated with alum or with your compound, in this case, drug 2. Prime the mice intramuscularly on day 0, boost the mice on day 14, and then we bled at day 28 to look at antibody titers. And just looking at the data over here, you can see that in protein alone, we do get some IgG production. These are anti-NP specific IgG. However, alum, which is the most commonly used control in this system for us at least, gave a nice increase in IgG production. And similarly, drug number 2 was able to have an adjuvant effect here and increase IgG production, much like alum was. I would point out here that what we've done is done this pretty quickly. So we haven't actually formulated drug number 2 for the purposes of this experiment. It was just combined with PBS and the nucleoprotein. And so we're interested in following up on these findings now and really properly formulating this drug to either decrease the amount that would be needed or even increase its efficacy as a potential vaccine adjuvant based on these early promising results. We do see throughout the system, rather intriguingly, a number of chemotherapies, specific classes of chemotherapies jump out for their ability to engage the innate immune system for some similarity to vaccine adjuvants or their ability to induce cytokine production. And what's interesting about this is that many of these chemotherapies were really primarily developed in immunodeficient mice. They were developed to be cytotoxic or to limit cell proliferation. And so most of the focus was on their effect of cancer cells, and many have not been examined in a mouse with an intact immune system. And so we've taken some of the more interesting classes of compounds and moved them into syngeneic tumor models where mice do have an intact immune system to see if we can get efficacy with these compounds. We have some early signs of efficacy, and obviously I want to stress these are chemotherapies. One would expect to see this. However, we're doing follow-up studies now to really see what impact these chemotherapies have on intratumoral immune cell infiltration, with the belief that basically the drug is doing two jobs. It's having its anti-cancer effect, but also eliciting key forms of inflammation. And we're hopeful that these things will combine nicely with things like checkpoint blockade and synergize through that secondary activity of engaging the innate immune system. So in conclusion here, we apply computational methods to high content imaging of PBMCs and construct these models to discriminate between innate immune signaling agonists. This can be a vaccine adjuvant or a more directed PAMP, for instance. But it goes beyond just discrimination. We're making associations between these compounds with each other and with our experimental compounds that go into the screen. And it's really this pairing of these imaging models with these other features, whether it be features in the images or within the proteomics, that we use to really gain a deeper understanding of the biology of these compounds and hunt for compounds that we believe will have some of these properties and abilities to induce inflammation. And we've applied this to at least one high throughput screen that I've shown here and shown that we can pull out a few compounds that induce inflammation. And we believe these have potential applications as vaccine adjuvants and potential immunotherapies. And just to comment a little more broadly about the company, my focus here has been on human PBMCs, but we perform similar analyses across a variety of cell types, both immune cells and non-immune cells. I've done quite a bit of work with human fibroblasts or murine myoblasts, and can even perform similar analyses on histological samples. So taking slides from various tissues and building models of disease or aging. And we've actually built tissue aging clocks here, so we can look, for instance, at mouse skin and make a prediction about the age of the animal from which the skin came from. I want to end by thanking the broader team. As I said, it's a pretty close-knit collaboration between the scientists and the computational experts. And really, I wouldn't be able to interpret this data or achieve anything without the help of my computational colleagues. I won't go through everyone's name individually, but everybody listed here touched some of this work in some way. And then finally, I want to leave you, if you'd like to receive a copy of the slides, you're certainly welcome to. And we have some additional online resources that you can click through here, one of which is a blog post with a sort of an interactive walkthrough of that mega map to give you a clearer understanding of how we use that software to sift through compounds and look for interesting hits. And then finally, we have a preprint available where we've looked for compounds to alleviate some of the defects in antiviral immunity as we age. Very similar experimental setup, just a slightly different take. It's certainly an interesting read if you have the time. So I think I will stop there and answer any questions that you might have.
Great. Thanks so much, Matt, for a really clear and compelling presentation. So I see that we have at least one question in the chat, and then we'll open it up more broadly for questions. But the question I see in the chat comes from Bob Shoemaker, who asks, what libraries do you plan to screen next?
Yeah, that's a great question. So, we decided to start with a repurposing library because we liked having that sort of anchor of annotation of knowing what each compound targets. But we have a keen interest in expanding this to new molecules. So one could imagine going after, say, a structural library. And in many ways, I think that has the potential to be more rich for innate immune agonist. One of the things about using a repurposing library is that these drugs have been used in the clinic. So if they induce systemic inflammation, they're not going to get prioritized for use. So it's actually really probably an unlikely space to find a lot of innate immune activators. We found a few, and in particular chemotherapies, where I think some of those adverse events would be better tolerated. But we would love to take this into more targeted libraries, things that, iterate on things like R848, et cetera.
Great. Are there any other questions from the audience? Wolfgang,
Hi, Matt. Really great talk. I know I said this before. This is a really exciting, very powerful technology. Basically, it becomes more and more powerful the more adjuvants you have screened and the more profiles you have to compare to. So how do you actually work with outside partners? So if somebody says, profile my adjuvant, what do they actually get in return? Are you able to give them profiles, not just of their adjuvant, but tell them this is how it fits in with others?
Yes, absolutely.
That's
Yeah.
the space where it's...
Yeah, absolutely. I mean, certainly we can show comparisons to sort of the data we ourselves have generated. And a good example of that is the partnership that we've entered into with the Bill and Melinda Gates. And so there we're taking adjuvants that the foundation is particularly interested in. And you're absolutely right. One of the strengths of this is that basically the data snowballs. So we're able to compare everything back to all of the data we've generated. And so what we can, for instance, give the Gates Foundation is a comparison of the adjuvants that they're interested in to the things that we've already run. So ASO3, IMA59, ASO1. We could do this likewise with any partner as we're doing the runs, fold in additional adjuvants. I think the only thing we couldn't show you is if we have a proprietary adjuvant from the Gates. Obviously, we can't tell you information about that, but certainly the things that are out there and available, we can share that data and explain those relationships using this type of phenomics.
As you work talking about the comparison, what I realized is how useful this technology would be to characterize and kind of validate mimics of adjuvants. So as you probably know, we're interested in mimics. The foundation has looked at mimics, but unlike a drug, it's not going to be a biosimilar, so it's a functional mimic. And one aspect of this comparison that everybody, I guess, is automatically going to be struggling with is to prove that what you're inducing is really the same kind of response. And the more, the broader the characterization of the responses, the more you can make that argument that it is really a mimic of that established adjuvant.
Yeah, I think you nicely highlight sort of the strengths and sometimes the frustrations of phenotypic drug discovery. Because the final readout is a phenotype, you could arrive at that phenotype through totally independent mechanisms. And so it's very important to design orthogonal assays that can capture that it's doing what you want to do. I think the kind of simple mouse vaccination model was one example of that, a pretty simplistic readout of IgG production. But we don't think this is going directly through, I want to be explicit, it's not going through a TLR pathway. It's not acting through the typical receptors. It's just that the final output and the inflammation gives you that output of increased inflammation, increased antibody production.
Yeah, one follow-up question. So when you're talking about mechanisms, have you considered using this approach to get first insight into potential mechanisms of action of a novel compound? So if you pull something out of a drug library that seems to be immunostimulatory, you have no idea what receptor, what mechanism it's going through. But if you already have a database of profiles induced by TLR2 agonists, TLR3 agonists, and so on, you would eventually get a sense of what is the common profile of a TLR9 agonist that sticks out. Have you tried to apply these profiles to new compounds to get a sense of what pathway is it likely working through, because that would facilitate the follow-up confirmation? Now you take a reporter cell line.
Yeah. So I think at this stage, I can't think of a precise example, but I think what you're highlighting is what we're hoping to do. Again, the data continually grows. The model is continually fed by the data that we acquire. So the hope is that the more we plug into the system, the more diversity and mechanism we're going to capture there. But I can't think right now a precise example of what you're highlighting, but I think it's definitely possible.
Thank you very much.
Thank you.
Yeah, that was a great discussion. I had similar questions about mechanism. I think those were addressed that discussion. So there's a few more questions that popped up in the chat, so we can go through these. So we have a question from Milton who asks, can phenomics help identify adverse events of adjuvants? And I'll add to that, just general reactogenicity. Can you terminate anything about this from your technology?
Yeah, we're certainly asked this question a lot, and I think what you ultimately want there is relationship back to clinical use of the adjuvants. And so I don't think we quite have that yet, but I do think features could be associated with adverse events. I think I pointed out IL-6, people frequently use this as something that could be an indicator of adverse events. We're quite good at different forms of cell death. So if you want to penalize certain forms of cell death, like pyroptosis, for instance, things that you might deem more inflammatory, that's certainly possible. I just don't think we have the full data set from discovery through to clinical translation to say definitively that I can point to key features. But again, the hope is that we'll get there.
Great. And then Amy asks, how do you account for potential differences in adjuvant effects when combined with different immunogens?
Yeah, that's a great question, and something that we're interested in pursuing with the Gates Foundation as well. And this initial iteration, we were sans adjuvant, but the plan now as we're moving forward is we're going to be combining adjuvants plus or minus antigen. And so basically there we can begin to understand that, if you put a particular antigen with a adjuvant, do you change the phenoprint that you get from it? What effect does it have there? But we're just now beginning those studies.
I think that'd be really interesting to see. So we have a few questions from Angeli so to start with, have you noted any age-specific impacts of adjuvants based on your phenoprints?
Yeah, so we've looked more extensively at PAMPs in terms of age-specific differences, and definitely when you use larger donor sets with patients over the age of 60 or 65 and patients middle-aged, you typically see, in the case of most PAMPs, increased cytokine production. So a kind of a reflection of that inflammation that occurs as we get older. And this likely derives from changes in monocyte phenotypes, which we also see evident in the morphological data. We haven't done that fine a characterization yet with the more complex vaccine adjuvants, but I would expect, well, we should do the experiment first, but I'd be curious to see if you see those similar changes. So definitely we think age impacts this, and that's something we're focused on.
And then has phenoprinting been applied to lymphoid tissue-derived immune cells?
Yes, that's a great question, and something we would love to do. We can handle tissues and certainly build models on them. It's just not something that we've done yet. On the mouse system, one of the things we have been interested in are things like thymic involution. So we've taken samples for histology from thymus, for example, to look at it, but it's still early days for those models. And the focus of that is really more aging, not necessarily looking at the influence of adjuvants or vaccination.
So as both Angeli and Milton kind of ask a similar question, have you looked at combinations of adjuvants? And I'll just give some background that, NIAID and I think some other, and plenty of our people are interested in the combinations of adjuvants and how those differ from the adjuvants on their own synergistic effects and whatnot. So do you have anything to comment on about that?
Yeah, unfortunately, we haven't done the experiment yet, but I think that's an intriguing idea. And even when we started, I think one of the thoughts was even just taking some of the more defined, PAMPs, TLR ligands, small molecules, and trying those combinatorially to see what effects they have. Because of the throughput of the system, it lends itself to that. We just haven't done the experiment yet.
Interested to see that. Are there any other questions for Matt? And feel free to unmute and ask them if you have them. So I'm not seeing any. Oh, it looks like Wolfgang's got another one. Go ahead, Wolfgang.
One more question. You mentioned antigen specificity of T cells, that you have not looked at this yet. Have you considered adding tetramer staining as one of the parameters that would allow you to see number of T cells, antigen-specific T cells, and their interaction with APCs?
Yeah, this is, again, it falls in the category of kind of on the list of things to do. It's something we've thought about. We hadn't thought specifically about tetramer. One of the interests I have is basically, could you take something like viral reactive T cells that have been expanded and basically combine those into the assay so that you could now actually look at and query APC T cell interactions. I think this is obtainable because we actually started working more in depth with T cells recently. I didn't have time to talk about that, but even using morphology, we're building models to discriminate CD4s and CD8s. And we're actually looking at things like checkpoint blockade. So basically our chops for dealing with T cells proliferating and working in our system is increasing. That's the way I think I would approach it. At least that's the way I've thought about it, is use something that's viral reactive, an expanded cell population. I think the problem is I would really love to see naive T cell priming, and I just don't know of a way to capture good human naive T cell priming in a dish, at least yet.
CD69 staining?
Oh yeah, definitely would work. Yeah. And we do a lot of model building. So I showed CD86 for the FAC staining. You can also include something like that, say, to look for APC maturation as well. We haven't done CD69 specifically, but I would think it would work in similar fashion. And one of the things that's been exciting to me is we can basically predict stains that aren't there. So by using the cell painting assay and incorporating some stains, you can actually build models that will infer whether or not something like CD69 or CD86 was there based purely on the other stains, on the morphological stains, which is a pretty exciting area. It doesn't work for everything. It has to, basically it has to somehow fundamentally be related to a morphological change in the cells. But this is something we're interested in doing, because when you do models like that, again, it's additive. You can always apply these retrospectively to your data. So we can go back through all the screening data we have and look for something like that if we can build a robust enough model.
I just have one comment about what you said about excluding chemotherapeutic drugs that had previously shown reactogenicity, strong inflammation. I wouldn't necessarily exclude those if they were used systemically and had side effects. I wouldn't exclude them from consideration as adjuvants, because the format is going to be very, very different. The dose, local application, formulation, and so on. So there might be something there.
Yeah.
Even if you gave a kilo 7.
Yeah, we agree with that. Yeah. Formulation is going to be the key there. And I think my point there was more that definitely if you look at the things that have adjuvant-like activity, there's a large representation of chemotherapies. And so I've often wondered if things in other classes where those adverse events wouldn't be tolerated, if you're making some sort of metabolic inhibitor, it's just never going to make it into our library. So we would have never seen, potentially other classes of drugs that could be immunostimulatory. But no, that's a great point. And I agree completely that the location is everything, especially for adjuvant formulation.
See, Milton has his hand raised.
Yes, hi. Thank you so much, Matt. Great presentation. Out of curiosity, can you describe a little more your experiment? What's a typical experiment looks like in terms of how many plates you handle at a time, how long it takes to prepare and analyze, etc. The cultures, a little more details about the experiment, just out of curiosity.
Yeah, so I can get back to you with precise numbers, but basically the simulations and cell culture conditions as we showed them today, these were thawed PBMCs plated overnight. So the actual stimulation is pretty short. That's a day. The staining itself takes hours. The imaging acquisition can take about a day. I'm blanking on precise throughput off the top of my head, but typically we're talking thousands of compounds at a time. So many 384 roll plates. I'll have to get back to you with a precise number. I just don't want to throw something out that's wrong.
Thank you.
But I believe the entire screening campaign, if I'm remembering correctly, for the 10,000 compound library, this was run in about 2 to 3 months, if I remember correctly. So it was run fairly quickly.
Okay.
All right. Any last questions? think we had a really nice discussion there. So thanks to everyone for the questions. Thanks to Matt for answering them. I think we can go ahead and end this this seminar now, but I'll finish just by thanking Matt once again for the excellent presentation, and reminding everybody that if you want access to Matt's slides, just send me an email. And if you want to be connected at all, just you can go through me, or I guess contact Matt directly. But thanks again, Matt.
Thank you all. Thank you for the opportunity.
All right, everyone have a great day.